Welcome to the Seed Germination Lab Experiment. In this investigation, we will observe how bean seeds begin to grow and develop into young seedlings. The aim of this experiment is to observe the stages of seed germination and identify the conditions that seeds need to begin growing. For this experiment, we need 3 bean seeds, a transparent container, and clean cotton wool. We also need water, a dropper, and a tray to hold our container. Finally, grab a label, a marker, a ruler, and an observation notebook to record your findings. The transparent container allows the roots and shoots to remain visible as they grow. First, place clean cotton wool inside the transparent container. The cotton wool should fill approximately half of the container. It will hold moisture around the seeds during germination. Next, place 3 bean seeds between the cotton wool and the side of the container. Space them apart and keep them visible so that each stage of germination can be observed clearly. Add a small amount of clean water using the dropper. The cotton wool should become moist, but it must not be flooded. Too much water can reduce the oxygen available to the seeds and may cause them to rot. At this stage, no roots or shoots are visible. Label the container with the experiment name and starting date. Place it in a warm, bright location with indirect light. Check the cotton wool every day and add a little water if it begins to dry. On the first day, the seeds are moist, but no root or shoot is visible yet. After absorbing water, the seeds swell. The seed coats begin to split and a small white root called the radicle emerges. The radicle is usually the first structure to appear. As germination continues, the roots grow downward and absorb water. The shoots grow upward toward the light. The young shoots gradually become green. By about the 7th day, young seedlings are visible. The roots are longer, the shoots have grown upward, and the first leaves begin to open. Observe the seeds at the same time each day, measure the root and shoot with a ruler, and record the results in an observation table. Please note that actual measurements may vary depending on individual seed growth and laboratory conditions. The experiment shows that seeds need water, oxygen, and a suitable temperature to germinate. Water activates the seed, oxygen is required for respiration, and a suitable temperature allows chemical reactions to occur. Test your knowledge on how bean seeds grow into seedlings. Read all 3 choices carefully, then choose the best answer. What is the main purpose of a seed germination experiment? A. To observe how seeds grow into seedlings. B. To measure the weight of seeds. C. To identify different types of flowers. The correct answer is A. To observe how seeds grow into seedlings. Which condition is necessary for most seeds to germinate? A. Dry soil. B. Water. C. Bright light all day. The correct answer is B. Water. What is the first visible sign that a seed has germinated? A. A flower appears. B. The root radicle emerges. C. The fruit develops. The correct answer is B. The root radicle emerges. Why is it important to keep the amount of water the same for all seeds in the experiment? A. To make the experiment fair. B. To make the seeds different colors. C. To increase the number of leaves immediately. The correct answer is A. To make the experiment fair. Final challenge. Which observation shows that a seed has germinated successfully? A. The seed coat remains dry and unchanged. B. A small root and shoot begin to grow. C. The seed becomes smaller without any growth. Take a moment to think. Then, write your answer in the comments using only A, B, or C. The correct answer will not be revealed in this video. Comment your answer below using A, B, or C. If you enjoyed this quiz, please like, share, and follow for more biology quizzes and experiments.